Welcome to Unit 3, Lesson 3. Today we're going to be talking about transcription, which is part 2 of a 3-part story. Last day we discussed part 1, which was replication. By the end of this lesson, you should be able to describe the structure and function of 3 types of RNA. We're going to do this in part 1 of the video, which is mandatory. And then in class, or video if you're away, we will discuss the process of transcription, including why, how, and who, and we'll be able to explain the purpose of the promoter site, initiator site, and termination site, be able to describe exons, introns, and alternative splicing, and finally be able to explain the difference between immature and mature mRNA. So to start with, we need to recall the 3 types of RNA. We went through this very quickly in the very first lesson on nucleic acids, and we just want to make sure we're clear on what they are. So first of all, RNA, recall, stands for ribonucleic acid, which is a polymer of nucleotides, and in this case a nucleotide has a phosphate backbone attached to a ribose molecule, which is distinct from DNA because it has an OH there as well as the OH there, so the extra oxygen, and a base, and these bases can be adenine, guanine, uracil, and cytosine. So we don't have in RNA thymine. The U replaces the thymine. Okay, so that's just a quick review of ribonucleic acid and the key components or differences compared to DNA. From this, we build up 3 different types of RNA. Your first type is mRNA, which hopefully you remember stands for blank RNA. That's right, messenger. Okay. So if you remember or recall, we just talk about DNA for a sec. Recall that DNA codes for thousands of genes per chromosome. And this is essentially the good copy of the blueprint. The original, the one that you want to protect and keep safe in the nucleus. It contains all the plans for all the proteins that could possibly be built by a cell. What mRNA does is mRNA carries the code, so it carries the DNA code or message. This is why it's called messenger. For a single gene, from the nucleus to the cytosol of the cell. And many of you maybe heard the word cytoplasm as opposed to cytosol for the cell before. The cytosol is just all the jelly stuff that the organelles of the cell are located in. So it's everything outside of the nucleus to the membrane that is jelly-like but not actually an organelle. Okay. mRNA is typically linear, we discussed last day? And is it double-stranded or single-stranded? That's correct. It's single-stranded. And it looks something like even this. You might have something like AUG, CAG, CUA, a string of nucleotides all strung together in a nice straight line. So one nucleotide joined after the other. And I've just put the spaces in for ease of reading. They really wouldn't be there if we looked at a three-dimensional mRNA strand. Then next we have our second type of RNA, which is called tRNA. Let's see if you can remember what that stands for before I write it down. Blank RNA. And hopefully you came up with transfer. And it's called transfer RNA because its job is to transfer the appropriate amino acid Remember, we're going to abbreviate amino acid as little a, little a. So the appropriate amino acid to a growing chain of amino acids according to the mRNA instructions. So it's actually going to build the protein according to what the mRNA blueprint says. Okay. We can recognize its shape because it's actually T-shaped, which is somewhat fortunate. It's not why it's called tRNA. And it is single-stranded. So it's one strand that folds back on itself to form a T shape. So I'm just going to draw this over to the side. A tRNA will look, actually I'll draw it below so we have more room. A tRNA kind of looks like it has a T shape. And at the top, it will attach to an amino acid. And the reason it has its T shape is because it'll have base pairs within itself. So it folds back and pairs. The A would have a U, the U with an A, and the G with the C. So it would base pair here. Remember, 2, 2. Whereas these little knobs off of it are just places where there aren't base pairs. There's not a complementary base pair to fold back with. So the top would have complementary base pairs. The bottom would have complementary base pairs. And then at the very bottom, you have what's called an anticodon. And this would be the anticodon. which complementary pairs with the mRNA. So this is how it knows which amino acid to bring in where. If we had a nice mRNA down here, it's 5 prime, 3 prime. The A would pair with the U, the C with the G, and the G with the C. And only will it bring in the right amino acid. ACG can only carry a particular amino acid, and it will bring it in according to the mRNA code. Okay. And then our last form of mRNA that we need to know about, there's interfering RNA, silencing RNA. There are different types, but you won't need to know about them in grade 12. The last type you need to know about is the rRNA, which stands for, think back to 2 days ago, 2 lessons ago. That's right, ribosomal RNA. So it is RNA that, along with protein, constitutes or makes up, is part of the ribosome. Which, if you remember back to Unit 2, we discussed when we make a peptide bond, the ribosome is the enzyme that forms peptide bonds between amino acids. And it is what we call globular, and once again single-stranded, or SS. And similar to how the tRNA folds back on itself, it folds back on itself to make this sort of spherical or globular shape, a big glob. But it's very specific. Every rRNA, if you pull it apart, it would go back into the same shape depending on its intramolecular bonds. Okay, the bond pairing between its bases. Okay, so that we're just going to finish right there as the end of part 1 of the video, the mandatory portion, and we will pick up the rest in class. If you have any questions, come ask in class or come see me. I hope you have a good day, and I'll see you in class. If you're away, you can continue on to the rest of the video. Have a good day.